with the next session, which is on CADASIL genetics and pathology. So I'm Bo Norvig from Lund, Sweden. I'm extremely pleased to introduce the next speaker, which actually needs very little introduction because she is one of the core people in this business, Elisabeth.
So thank you, Bo, and it's really a great pleasure, 20 years later, to see many people that were at the beginning of the story, and it's really a great pleasure. And I think that was an idea of Marie-Germaine to organize this 20 years after meeting, and I think that is an extremely good idea, and also in this place, which is wonderful. I mean, it's really... So I'm going to try to go to the prehistoric, but my talk, in fact, would be divided somehow in two parts. One is going to be the prehistoric, because we have to really define clearly what is CADASIL. And after that, I will go on a more recent work trying to sort out what are the consequences of non-CADASIL mutations in the Notch3 gene, because we have to avoid confusion for the patients, confusion for the future trials, confusion also, we have to know what is the evolution of these diseases, which, if they are not CADASIL. So that's would be the essential part of the talk. Next slide. Ah, c'est moi, d'accord. Okay, so that was prehistoric but very important stuff. And I think that this established somehow what is really CADASIL. And CADASIL was defined first, we have to characterize, and Hugues presented you what's the phenotype of this novel disease at that time, to realize that it was genetic and it was autosomal dominant. Marielle Baudrimont has this seminal contribution showing that one of the markers of this disease was the presence of this granular osmophilic deposit in the basal membrane of vascular smooth muscle cells. And this is a very, very specific marker. After that, but I will not come back because I think Anne will talk about it, on another marker in the skin. This is a skin, This is, in fact, cis gum. You can find them in somehow all vessels, and particularly in the vessel of the skin. And so this is characteristic and very helpful. But there was also another marker in the skin, which is the accumulation of the extracellular domain of Notch3 that you can detect by immunohistochemistry. And after that, there was a linkage. In fact, thanks to these first large families that Marie-Germaine talked about, in fact, we were able to map the locus of CADASIL on the chromosome 19. And afterwards, Anne in the lab identified the gene, which is the Notch3 gene. So these are really markers. As you will see, we cannot say that clinical and MRI features of CADASIL are specific of CADASIL. There are some features that are more specific, like the temporal lobe involvement, but I would say that it's very difficult when you receive patients in the diagnostic lab on the MRI to make sure that's going to be a CADASIL patient. The EM is a very good marker for sure, but it's a lot of work. The linkage is good also, but the main thing is now to have access to the gene. So, in fact, just because it's the first time that in the seminar, in the workshop, I would just spend a few minutes on this particular gene. It's a 33 exons gene encoding for more than 2,000 amino acids. It's transmembrane receptor, and it's this receptor here. You have the full-length receptor, and it is characterized by the fact that all these little green boxes here encode for EGF-like motifs, which contain 6 cysteine residues, and the two blue one also, but these two blue one have a function which is to bind the ligands of the Notch receptors. And this is a transmembrane domain, and you have here the intracellular domain. But this full-length receptor will be processed and will be addressed to the membrane after several proteolytic cleavage episodes, and you will have here the extracellular domain and the intracellular domain. And this part here, when cleaved completely here, after the second and third cleavage, will go to the nucleus and have a role in transcribing genes. So this is a gene. And what do we know? So what do we know? And I would be extremely precise. In fact, as at the beginning, I mean, to define what is CADASIL, in fact, we used 50 index patients that were included in the story, in the study, and these patients have an ischemic familial small vessel disease, and at least more than 30 of them were either linked to chromosome 19 and or having GOM on skin biopsy. And this, I think, is quite important. And so, based on this, we in fact screen all 33 exons of Notch3, and as you see, this was a prehistoric period because sequencing was not easily available, so we have to use strategies that were not as sensitive as sequencing. And once this methodology, I mean, pull up a variant, we will then sequence only the variant. 45 of these index patients were heterozygous for a missense mutation that was leading to an odd number of cysteine residues, and within one of these EGF motifs. So this was extremely already striking. But as you say, but still, there were 5 index patients, have no mutation, so how come? And in fact, two of these patients, in fact, were typically CADASIL patients because one is GOM and the other one was coming from a linked family to chromosome 19. So, so in fact, what happens is that these people, these two patients, have been resequenced recently, in fact, both in Italy and in France, and in fact, both of them had a cysteine mutation that was missed by the previous approach. And there are still 3 patients, but I think that these 3 patients, from the information that I have, may not be CADASIL patients, because it's not GOM for the moment, discovered in these patients. And so we are now, I should say 47 patients out of 47 that were suspected of CADASIL have this heterozygous missense mutation. And this is one of the EGF, the opposite side, and you will see that either they get an additional cysteine within the motif, or they lose one of the cysteine. And that was published in Lancet. And this, for me, is somehow the definition, the molecular definition of CADASIL, but it was very important to other groups, and Dishkan's group and Kalimo's group, and in fact have conducted studies starting with proven CADASIL patients by EM, and here Kalimo's groups, 131 proven, also 131 as an odd number of cysteine residues. In the 125, we have 120. And what was interesting in this study is that it was not only missense mutation. 95% of the mutations are missense mutations, but 5% are either deletion and whatever mechanism, but they all lead to an odd number of cysteine residue. So that was, so now I think that we can say that all mutation in CADASIL biopsy-proven patients lead to an odd number of cysteine residues. And somehow that should be the definition of this disease. So now, where are the mutations? And in fact, This again were on several series from Italy, from Germany, and the series that I was talking about, and nothing has changed, in fact, between these three. You see that most cases, the mutation are located within exon 4 and exon 3, which encodes the EGF 1 to 5. And this is extremely stereotyped mutation, and you see that there is no mutation here starting at the 25. There are mutations now in 24, etc. There's something interesting. There's one exon that never has mutation. And I don't know why, but this is exon 16, and even in extremely large series now, you see no mutation. Okay, so now the title that Marjamin asked was molecular genetics and genetic counseling, so we have to talk a little bit about the diagnostic of these patients. Why screening patients for the NOTCH3 mutation when a patient has a small vessel disease, and how to screen these patients. And obviously, the importance of screening those patients is to avoid misdiagnosis. I look back to the former data and in fact realized that 30% approximately of initial CADAGIL patients that we had in 1990s were in fact diagnosed as having multiple sclerosis, a number of other psychiatric disorders, and these patients, for example, with multiple sclerosis, they have immunosuppressive treatments, etc., and which obviously was not adapted to these patients. So I think it's important to avoid misdiagnosis or no diagnosis. It's important also for genetic counseling, and I will come back to that a little bit later on, but to do a real, correct genetic counseling and diagnostic, you need a highly sensitive and specific test. And the problem is that between 1993 and 2003, that was a prehistoric period. And in fact, 1993, the gene was not identified, so the only possibility was linkage to chromosome 19. In 1996, the gene was identified, but sequencing was very expensive and difficult. And so what we and others, in fact, performed was a sequencing of exons 3 and 4, and EM skin biopsy. And only the patients with EM skin biopsy positive and no mutation in exons 3 and 4 would be sequenced for the rest of the exons. And 2001, Anne also showed that it was very sensitive, for the diagnosis to have a skin biopsy immunostaining with the monoclonal NOTCH3 antibody. But this requires, if we want to be efficient, frozen material, and it's not very, they're not very simple now. In 2003 and from 2003 to 2013, sequencing, arrival of capillary machines, and this changed our life. And so now, in fact, I will talk about our experience since 2003. And so we talk about the last 10 years and not the last 20 years. And so what happened during 2003 and 2013? In fact, we are the reference lab at the national level for the diagnosis. So all French patients suspected to have a CADASIL the samples come to the lab, and we receive also a number of samples from outside. But between this, during the last 10 years, we received about almost 3,000 patients. And these patients were referred for either lacunar infarcts and leukorrheosis or cognitive defects and leukorrheosis. They could be familial or sporadic. I have to say that I realized that there was something strange when I prepared this presentation, is that I realized that among these almost 3,000 patients, there are 43% men and 57% females, which I don't understand. There is somehow a bias in the referral, and these patients have a mean age of 55. And when I look to the CADASIL patients, because we identified in this consecutive cohort 13% of patients with CADASIL patients, and again the proportion was the same, meaning 43% of males and 57% of females. So maybe the males don't go to the hospital, I don't know, and they are not as precocious with their health, I don't know, but this is very significant, and I don't know why. So I would say this is kind of interesting because 13% of patients referred for a suspicion of a small artery disease are showing a typical cadazine mutation. This is one of the highest proportion than you would imagine in genetics lab when you screen for, you know, the phenotype as was shown by Hugues de Marco. I mean, there are many sporadic small vessel disease. So I think it's a very important proportion. But what to do after that once the diagnosis has been established on the basis of an odd number of cysteine residues? In fact, as I say, it's important to avoid misdiagnosis, and it's also important to avoid long and costly investigation in the affected relatives of the index. But I would like to show you that, in fact, during the same period, we have 361 index patients, the first patient to be diagnosed in a family. And if you look here, we have only 208 relatives. If you relate that, you can relate that to the approximate size of a French family, and it's autosomal dominant, so you would expect half and half. So, in fact, it fits, and it fits. So now, maybe the question I would answer some questions. And now, what about once we have this diagnostic test positive? What about genetic counseling? Genetic counseling, we are not in a problem of prenatal diagnosis. I mean, in CADASIL, within the last 20 years, I got only 2 requests for prenatal diagnosis. And within these 2 requests, none of them, the couples, did not go further and were not performed. It's a disease that starts at 50 years old, and we hopefully, thanks to the work of Joutel and the Leduc network, we'll have a treatment. And so I think that there is no reason for such a late disease to have prenatal abortion. But still, it's available. So the real question is a pre-symptomatic testing. And this should be extremely cautious. And in France, we have, for severe disease with late onset and no treatment, in fact, the patient, it's not a patient, it's a member of a family, should benefit from experienced multidisciplinary teamwork, and there is a multi-step procedure, so the members can think about it, make sure that they want this test or they don't want this test. And so in La Riboisière experience, 2007, 2010, and this was a work that was published by Sonia, yes, and Annie Kurtz. In fact, you see, we had only 33 asymptomatic members who came to the facility to ask for pre-symptomatic testing. 22, in fact, decided not to go further and not to perform tests. So it was good that they have this opportunity to think about it and not having testing. 6 were mutated. And what I would say is that it's very important for providing care before, during, and post-testing. If you do that in this short series, in fact, there was not big damage, but it's a short series. So now, the next step. 361 CADASIL patients, but we have also in this cohort 2.8% of patients who have a rare missense mutation within NOTCH3, which does not affect the number of cysteine residues. And when I say rare, I mean mutations that are completely absent from databases, or at least have a frequency which is below 1 in 2,000. So what are these rare missense mutations doing? Are they polymorphisms or are they deleterious variants? So, and it's important, in fact, in the literature, there have been several reports, saying that these arginine, 75 proline, whatever, are in fact missense variants causing CADASIL. Or sometimes people say most likely causing CADASIL, but not sure. And in fact, most of these variants now, we know that they are not causing CADASIL and they are polymorphisms. Why we know that? There is a huge progress in the databases, and now we know that some of these variants are very frequent, and their frequency is not compatible with the frequency of CADASIL. And also you will see, in fact, some of those patients had no GOM, but it was not performed in each series. Sometimes they are said to have GOM, but when you look at the pictures, it's difficult, but Kalimo, I know, would have the best expertise on that. So, in fact, we decided to sort out this problem because we cannot answer to the questions of the clinician saying, okay, we have a variant, it's not in databases, but what is he doing? So what we did, in fact, and this is a project that we run with Françoise Chapon and Claire Debout at CHU de Caen, who are the pathologists in charge of all the EM analysis of the skin biopsies, and many people have been involved, particularly Maria Delmar, Amador, and people also of the diagnostic lab, and more recently, Carole Prieto. And in fact, we took, 19 patients that have a rare missense mutation absent from databases. To give you an idea, absent for databases means that the variant is not present in at least 15,000 people, but much more, because sometimes in database we have more people. And so these patients were sequenced for EGF2 to 24, no mutation leading to cysteine residue. And in fact, we performed systematically a skin biopsy, and the pathologists were blind to mutation status. As Anu would certainly say in a few minutes, in fact, it's extremely important that a number of vascular sections should be analyzed so that the test is very sensitive with good controls. And that's what was done here. And in fact, what are the results for the moment? We have 17 patients here who have no GOM. So these variants, who are essentially variants absent in databases, so candidate for the disease, in fact, no GOM, so these patients are not CADASIL patients. But we were surprised to see that 2 patients here have GOM. And to convince you, you can look here, and these are François Chapon and Claire pictures. You will see a GOM here, and you see this typical GOM here also. In these 2 patients here, and these 2 patients do not have a mutation leading to another number of cysteine residues. But very surprisingly enough, these 2 patients who are unrelated, completely unrelated, in fact, they carry the same mutation, which is a very rare one. So we are now, I'd like to say, okay, is this 2 hypotheses, this mutation causing CADASIL? And if so, what's the mechanism? And have we missed, on the opposite, have we missed a true CADASIL mutation? And this mutation is in linkage disequilibrium with the true mutation that we have missed. So in order to solve that, I have 3 hypotheses. It is really leading to CADASIL, and it's a missense mutation. The mechanism, what is the mechanism? No GOM, is it a conformational change? Is there an interaction of this amino acid with the component of basal membrane? I'm very cautious. It may well be possible. Science is always surprised. So that's possible, but we have no proof for it for the moment. But what can happen is that this variant, in fact, And this is known for missense mutation and stop codon. In 10% of them, they may lead to abnormal splicing of the messenger RNA, which is encoded by the allele. And what could happen is that there's an abnormally spliced messenger RNA leading to end-node number cysteine residues, and we are back to the classical CADASIL story. Another hypothesis is that this mutation is really, in tight linkage disequilibrium, in fact, with the real mutation, and we have missed the real mutation. Why? Because we have sequenced exons and, intron exon junctions, but there could be a very deep intronic mutation leading to abnormal splicing, or a deletion that if we have a deletion, we are sequencing only the normal allele. So we are currently sequencing the cDNA that would provide the answer to these two questions, and we do that with using messenger RNA from skin biopsy fibroblasts, and the Notch3 gene is clearly expressed, and there's no problem. And for the moment, I have to say that Carol is doing it for the moment, that we have not found any mutation in the messenger RNA, and the mutation is in the messenger RNA. And we have not also detected deletion so far. But still, we have a little part of the cDNA that we still have to sequence. So that's where we are right now. And so the important thing to say for clinicians is that if you have a missense mutation, please do a skin biopsy. In 90% of the cases, you're going to be eliminating a CADASIL. And this is very important because I've seen different patients that were convinced to have CADASIL, and in fact, they had polymorphisms. So that is important. But in some cases, maybe 10%, we don't know, maybe just this G73R, they have GOM, and we have to go further. So this is a very practical, there's very practical, consequences. So now back to the story. In fact, we have also, when we screen these patients referred for Notch3, we have found 9 index patients. You see, it's very rare, that do not have missense variants, but have stop codon variants. And stop codon are, in fact, mutations that, in most cases, lead to degradation of the messenger RNA, which carries the stop codon. So meaning that the allele that would carry the stop codon would just be degraded, and the consequence for the patient is that he would have half dose of the Notch3 protein, but the Notch3 protein, a normal one. So the main questions are those variants that in most cases would lead to haploinsufficiency deleterious or not? So the problem is that this is what happens in the vast majority of stop codon. So you would expect that in vast majority there would be half dose. The problem is that a few stop codon, like the missense, can lead to abnormal splicing. So again, 3 hypotheses. First hypothesis, a stop codon and an haploinsufficiency is not deleterious. And as Anne will certainly talk about it, in the mouse look okay until they are quite late, even if they don't have at all the Notch3 protein. So, but the mouse is not a man. So either it's polymorphic and non-deleterious, either it's deleterious and leading to CADASIL, exactly the same thing, by an abnormal splicing. Third hypothesis, these are deleterious variants causing haploinsufficiency, which lead to a novel small vessel disease phenotype, which has to be described. To solve and to clarify this hypothesis, again, skin biopsy. Skin biopsy would answer hypothesis, I've done a mistake. Skin biopsy would answer hypothesis 2 here, because if you have GOM, it's a CADASIL, yes, we have to find the abnormal splicing. the RT-qPCR of Notch3 messenger RNA from skin fibroblast or muscle could tell you if there is an haploinsufficiency. You can quantify the messenger RNA, and you can say, is there? But if you show that there is no, if you show that there is no GOM and that you have an haploinsufficiency, that does not mean that it's deleterious. There are many stop codons in each of you in the room, at least an average of 50 stop codons, heterozygous stop codons, which are not deleterious. At least you look normal. So that means that we have to prove. How to prove it? Two ways. Either you are lucky and you find a patient with a stop codon, you have both parents, none of the parents have the stop codon. That means that the stop codon appear de novo and the patient is sick and the parents are not sick. That's why I bother many of you in the room for several families to try to find the parents. But as these guys are referred at 55, generally one of the two parents is dead, so we cannot conclude formally. The other thing is co-segregation. If you have a stop codon, if it is deleterious, and if you analyze the relatives of the patient, you should be able to find, if it's not a de novo mutation, half of the relatives with a stop codon, half of the relatives with not stop codon. And which one are affected? If it is deleterious, you should have a co-segregation of the affected phenotype with a stop codon. So we decided to go and try to understand that. And so we have, on the whole period, we have in fact 12, because these are very rare, we have 12 unrelated patients with stop codon, and in 5 of them, for the moment, we have, it's not at all finished, we have known clinically affected relatives. So the strategy was to perform skin biopsy in 6, for the moment, we have not finished for the 12, 6 index patients, quantify the messenger RNA. This has been done from 2 index, but in muscle, and as muscle is a little bit invasive, we now know that we can quantify it using fibroblast, and so that would be easier. And clinical and MRI investigation of consentive relatives, and there are many people who have been involved in that, particularly Maria Delmar and Dominique Hervé at the neurology department, and genotyping of all consenting relatives. And to give you the first results, in fact, this is a typical CADASIL patients here, and these are 5 index patients, again, done in Françoise Chapon department, and none of the index patients have GOM. The second point is that among the 2 patients who have the head muscle biopsy with quantification of the messenger RNA, in fact, the messenger RNA which carries the stop codon is completely degraded, not totally. You can detect it approximately semi-quantitative method with 5%. So there is a Notch3 haploinsufficiency. So is this now Notch3 haploinsufficiency causing a disease or not? So this is an MRI of 8 symptomatic patients with this Notch3 stop codon. Some are index, and some, this, for example, is the father of this and index, and this is a sister of this index. And what you can see, but you have to realize that these patients are referred for Notch3 screening, so it's not very surprising for the index patients that they have MRI that are consistent with a small vessel disease. Although some of them are a little bit like this patient here, a little bit different, I would say. Here you have the index patient, 55 years old, and this is the father. And it's very interesting because the father of this patient, we got the DNA sample of this patient 5 years before her daughter. But the problem is that, you know, this patient was not sent at all for CADASIL. It was sent because he has a cerebral hemorrhage. In fact, it was a huge hemorrhage in the cerebellum. And he was sent for cavernous angiomas diagnosis. As we do also the cavernous angiomas, when we saw the name of these patients, we say, oh, come, that should be in the patronym dictionaries that we have a patient, and we realized that he was the father. And we claimed for the MRI, and you see this MRI here. But he was 72 years old, so the patients were not, the clinicians were not very, anxious about this, and in fact, it has the same disease. And this is a sister, this is a sister also, for example, of here with the same, basically, leukoencephalopathy. So, okay, but we need more, what about the evolution? Here you have the same patient several years later. Here, and décidément, il est un peu trop proche. Okay. So, in fact, here. What's striking me is that the evolution is not dramatic. Huh? For me, it seems that, in fact, several years later, let's look, for example, here, the F3 index patient. There's not that much change, basically. So maybe that this disease, if it is caused by an haploinsufficiency of NOD3, may have a slow evolution and a different evolution than CADASIL. What about segregation? We don't have enough families for the moment. This was done with Maria, and in fact we have now 3 more families in which the co-segregation has been done. And in fact, this is one of the largest family. The index patient was here, and her sister was affected, the sister also and dead, and this cousin also. And MRI has been performed in the second generation here. And in fact, 13 members have been genotyped, aged above 45, and have an MRI. And all 6 affected are mutated, all 7 unaffected are not mutated. We have 1 asymptomatic member, aged 42 years old, I will show you in a minute, with a normal MRI, also this patient is mutated. So it seems that there could be a co-segregation with a phenotype. It's the same in this family here, where the daughter and the father are affected. This cousin here is unaffected and does not carry the mutation. So possibly there is a co-segregation. We have at least 2 additional families with the case also. But to show you, we have to be careful on the MRI lesion. Here is 1 member of this large family, 63 years old, not mutated. The MRI was considered to be normal. Here is a sister, and 56, and this MRI, which clearly is not normal. This is a 42 years old. Again, the MRI is not normal. But this is a 42 years old of this family, and there are little things, this little dot here, this is the dot here, but I mean, we cannot consider that this is affected. So we have to pursue and to make sure of, and we have the material now in terms of families. So the conclusion at this point would be that, in fact, CADASIL is a disease, and that's what we think should be considered, characterized by highly stereotyped mutation leading to a number of cysteine residues, GOM, and NOD3 extracellular domain accumulation. 10 to 15% of patients referred for molecular diagnostic, depending on labs, in routine practice have CADASIL, which is quite enormous. Importance of skin biopsy for rare missense variants not affecting the number of cysteine to sort out the rare CADASIL patients. Stop codon do not lead to CADASIL, but they may be causing a novel vascular leukoencephalopathy whose phenotype has to be established. Practical implications. Stop codon can be anywhere in the gene. So just to remind you, we are only sequencing the 23 exons encoding the EGF domains. So 10 exons more can carry stop codon. So if this is a phenotype of a disease, we will have to implement the sequencing of the 10 additional exons. I've not talked because I didn't have time, but I'm sure Anne will talk about it. Additional phenotypes by very rare NOTCH3 activating mutation affecting the heterodimerization domain, but these are very rare. 1 out of 725 patients tested. And so importance for good genetic care, good genetic counseling, future therapeutic trial to establish precise criteria for NOTCH3 non-CADASIL mutated patients. And my last slide is this one. In fact, is that, so next step is that this plus this plus this, 15%. So what do have the 85% remaining patients? So that would be for tomorrow with Dominique Hervé. And this, we have to work with you on that. And last but not least, all the patients. In fact, I introduced all the people who worked on the diagnostic part and once the gene was identified. But there are many other people who are in the room who have contributed enormously at the part of the clinical research step on the analysis of the patients, such as Katayun Vaidi, Valérie Bius, etc. But I just positioned myself just after the diagnostic, in fact. And so all these people, I would like really to acknowledge Françoise Chapon for the EM study, and Dominique and Maria has done also a very good job, and you all know her, and the support also. And I'd like also to acknowledge the Le Duc Foundation, because for the SADB, this is, I mean, instrumental, and also the structures in CERM and the APHP Hospital, Genetics and Neurology Lab. And I would just be pleased to take some questions if you have some.